Classification of enzymes. In this e-lecture, we will study about the different classes of enzymes made by International Union of Biochemistry and the Enzyme Commission number. Let us start with introduction. Many enzymes have been named by adding the suffix ase to the name of the substrate. For example, urease, which catalyze the hydrolysis of urea to ammonia and carbon dioxide. Similarly, arginase, which catalyze the hydrolysis of arginine to ornithine and urea. And third one is phosphatase, which catalyzes the hydrolysis of phosphate esters. However, this nomenclature has been impractical in many cases and trivial, non-systematic or chemically uninformative names have come into wide use. Because of this fact and because of the rapidly increasing number of newly discovered enzymes, a systematic nomenclature and classification have been adopted on the recommendation of International Enzyme Commission. IUB, International Union of Biochemistry. According to International Union of Biochemistry, the enzymes are divided into 6 functional classes based on the type of reaction which they catalyzed. In the IUB system, each enzyme has a particular name and a unique identification number. The unique identification number of the enzyme is called the Enzyme Commission number. The systematic name of each enzyme consists of two parts. The first part of the name of the enzyme represent the name of the substrate, and the second part represent the ASE, which specifies the type of the reaction. The second part ends with ASE suffix and it specifies the type of the reaction which the particular enzyme catalyzes. The recommended name of enzyme for everyday uses is often an enzyme's previously used name, that is the common name. 6 classes of enzyme. In IUB system of enzyme classification, all enzymes were categorized into 6 major classes based on the type of reaction they catalyzed. And these 6 classes are OXIDO-REDUCTASES, TRANSFERASES, HYDROLASES, LYASES, ISOMERASES, and the last one is LIGASES. Enzyme Commission number. These 6 classes of enzymes were divided into subclasses, and these subclasses are further divided into subclasses. Based on the classification, a 4-digit unique number called Enzyme Commission number is assigned to each enzyme as an identification code. Each digit in the Enzyme Commission number indicate a specific category in the classification. For example, the enzyme alcohol dehydrogenase, the Enzyme Commission number is 1111. The first digit of this Enzyme Commission number indicates the major class, for which alcohol dehydrogenase, it is the oxidoreductase. and the second digit represent the subclass. The third one or third digit indicate the subclass, and the fourth digit indicates the systematic specific name of the enzyme. And the systematic specific name of the enzyme consists of two parts. The first part indicates the name of the substrate, and the second part indicates the nature of the reaction which that particular enzyme catalyzes. For example, alcohol dehydrogenase, it belongs to the class OXIDO-REDUCTASES. The enzyme commission number is 1111. The first one represents the class of enzyme and it is oxidoreductase. Second one is the subclass of enzyme and here it is acting on the CHOH group of donors. The third one is a subclass of enzyme with NAD or NADP as a acceptor, and the fourth one represents alcohol NAD oxidoreductase. The systematic name for this enzyme is alcohol NAD plus oxidoreductase, as the substrate is primary alcohol. Therefore, its systematic name is alcohol NAD plus oxidoreductase. Now the 6 classes of enzyme. The first class is oxidoreductases. An oxidoreductase is an enzyme that catalyzes the transfer of electrons from one molecule, that is the reductant, to another molecule, which is the oxidant. Reductant is also called the electron donor and the oxidant is called the electron acceptor. This group of enzyme usually utilizes NADP plus or NAD plus as a cofactor. Example, alcohol dehydrogenase belong to this category. In this reaction, the A is the reductant, which is the electron donor, and B is the oxidant, that is the electron acceptor. Transferases. Transferases is a class of enzyme that enact the transfer of a specific functional group, example a methyl or glycosyl, from one molecule, which is the donor, to another, which is called the acceptor. The systematic name of transferases are based on the form of donor-acceptor group transferase. In the enzyme commission numbering system, transferases have been given a classification of enzyme commission 2. The third class of enzyme is hydrolases. Hydrolases are the enzyme that catalyze the cleavage of covalent bond by using water molecule. There are different types of hydrolases, which are esterases such as phosphatases that act on ester bond, and proteases or peptidases that act on amide bond in peptide. Some common example of hydrolase enzymes are esterases, includes lipases, phosphatases, glycosidases, peptidases, and nucleosidases. Esterases cleave ester bond in lipids and phosphatases cleave phosphate group. LYASES enzyme. A LYASES is an enzyme that catalyzes the breaking of various chemical bonds by means other than hydrolysis and oxidation, and it forms a new double bond or a new ring structure. The reverse reaction is also possible in LYASES. For example, an enzyme that catalyzes this reaction would be a lyase. In this reaction, the ATP is converted to CAMP and PPI. The lyases differ from other enzymes in that they require only one substrate for the reaction in one direction, but it requires two substrates for the reverse reaction. Another class of enzyme is isomerases. Isomerases are a general class of enzymes that convert a molecule from one isomeric form to another isomeric form. The isomerases facilitate intramolecular rearrangements in which bonds are broken and formed. The general form of the reaction is as follows. AB is converted into BA. There is only one substrate yielding one product. This product has the same molecular formula as a substrate, but they differ in their bond connectivity or spatial arrangement. Isomerases catalyze change within one molecule. They convert one isomer to another, meaning that the end product has the same molecular formula, but they have different physical structure. Enzyme Commission 5.1. The category Enzyme Commission 5 number is provided to the isomerases enzyme, and this Enzyme Commission 5.1 include racemases and epimerases. These isomerases invert stereochemistry at the target chiral carbon. The racemases acts upon molecule with one chiral carbon for inversion of stereochemistry, whereas the epimerases target molecule with multiple chiral carbon and they act upon one of them. A molecule which has only one chiral carbon has two enantiomeric forms, for example serine, and it has isoform D serine and L serine, whereas the molecule with multiple chiral carbon has two enantiomeric forms at each chiral carbon. Ligases. This is the last class of enzyme. These enzymes are capable of catalyzing the reaction of joining two large molecules by establishing a new chemical bond, generally with concomitant hydrolysis of a small chemical group on one of the bulky molecules, or they simply link two compounds together. For example, enzymes that catalyzes the joining of CO, CS or CN, etc. The common name of ligases often include the word ligase. For example, DNA ligase, it is an enzyme commonly used in molecular biology laboratory to join together DNA fragments. The other common name for ligase includes the word synthetase, because they are used to synthesize new molecule. For further reading, I would like to suggest a few books of biochemistry. First one is The Biochemistry by Albert L. Leininger, and a book of biochemistry by S.N. Gupta, and Fundamental of Biochemistry by Jain and Jain. Thank you for watching.